Hello, thank you for joining us for today's Meet the Author interview with NPP. I'm Sophia Hupalo, the editorial intern at NPP, and joining me today are Chloe Jordan, our special projects manager, and Carrie Martinowicz, NPP's social media editor. And today we're chatting with two authors, Amanda Elton and Monica Faulkner, who recently co-first authored an NPP paper called Acute depletion of dopamine precursors in the human brain, effects on functional connectivity and alcohol attentional bias. This work was conducted in Charlotte Bodeger's lab at UNC Chapel Hill, where Amanda Elton is a research assistant professor and Monica Faulkner obtained her PhD. Monica Faulkner has recently moved on to the National Institute on Drug Abuse, where she is a postdoctoral fellow in the Laboratory on Clinical Psychoneuroendocrinology and Neuropsychopharmacology. So check out the article in the video description and reach out to us on Twitter if you have any questions. And for our first interview question, I'll turn it to Chloe.
Thanks, Sophia, for the introduction, and a huge thank you to Amanda and Monica for doing the interview with us today. The first question we had for you is, which finding in your paper were you most excited or most surprised to find?
Well, for me, it was the findings from the mediation analyses and the fact that this study design actually allowed us to identify the specific dopamine pathways that contribute to attentional bias. It was just really gratifying to see that there were some commonalities in the findings across the three different tasks that we used, and specifically the connectivity between that frontal insular cortex and the striatum.
I would agree with Amanda. I think that seeing that the dopamine depletion method for me, that it actually works. And I am a cognitive neuroscientist, but I love behavior. So seeing those depletion effects across all of the tasks and looking at them differently with the current binge drinking versus the adolescent binge drinking, I thought was very interesting and really gratifying to see with that particular study design. So the behavioral findings and the neuroimaging findings and the depletion effect were really exciting for me.
Thank you. That's so great when things synergize like that. Our next question is, could each of you speak about the process of how you determined the manuscript would be co-first authored?
Yeah, I think it happened kind of naturally. So I was a graduate student in Charlotte Bodeger's lab. So I was learning neuroimaging during that time. And Amanda was a postdoc, so she was an expert in neuroimaging at that time. So during my entire PhD, I always kind of leaned towards Amanda once she got into the lab to help with the neuroimaging piece of my dissertation. So once I was finished, there were many questions and more analyses that we could do, and it just wasn't in a great place for publication. So once I moved on and, got my postdoc and started working here, Charlotte approached me and said, hey, Amanda's going to take a second look at the data, and we're going to see if we can get it in a place where it's ready for publication. And Amanda, with her expertise, she worked her magic and did these amazing mediation analyses. And we came up with the final manuscript. So it was only right that with the amount of effort that Amanda put in and that I put in with my dissertation that it was going to be a co-authorship. But what do you think, Amanda? How did the process work out for you?
Yeah, I just want to add that I felt very fortunate to be able to tag along at the end with this pretty unique study. Monica put in so many hours collecting each individual subject in the study. And so just getting to actually look at the data after all that was done was pretty exciting. Yeah, so I really appreciated getting involved.
Well, I appreciated you helping.
Thanks for talking about that with us. So I guess a question that we had is are there follow-up studies in the Bodeger lab that are going on to follow up on these findings?
Yeah, since I'm still there, I can speak to that. So in the paper, we suggested that the functional connectivity between that anterior insula or frontal insular cortex and the ventral striatum could be a marker of susceptibility to reward conditioning. And so we're currently following up on that hypothesis. So we have a couple projects that are happening in the lab right now that are exploring whether there are brain connections, specifically testing again the connections that we looked at in this study to see whether they could be altered by adolescent alcohol use, which is assessed retrospectively in adults undergoing an fMRI scan. And since we know that adolescent alcohol use can alter brain circuits, and adolescent alcohol use is also a major risk factor for future alcohol use problems, one potential brain mechanism of risk for alcohol use problems later in life could be through altered frontal striatal connectivity related to reward processing, or reward conditioning specifically. And so the studies that are going on in the lab right now are examining whether these specific connections are in fact affected by adolescent alcohol misuse, and whether in turn those changes result in increased bias toward non-drug rewards. And we're actually using a reward conditioning task that's very similar to the one that we reported in this paper.
Okay, and last, I think we wanted to talk to you about both of you, and so we were hoping that you could tell us a little bit about what kind of current projects you're working on that you find most exciting and how they fit into some of your long-term research interests and goals.
Yeah, so my long-term research interests lie in better understanding the neurobiology and neuroendocrine mechanisms of addiction-related behaviors, and using that knowledge to identify treatment targets and interventions that are translatable into community-based clinics, right, for alcohol and substance use disorder. So I'm definitely a translational researcher in the making. I would love to see some of our interventions in community clinics being used widely. And from what I learned from the study that Amanda and I published, as well as my current work in the lab of Dr. Lorenzo Leggio, I'm currently planning investigations to look at the interplay of the neuroendocrine system, specifically peripheral metabolism, nutritional interventions, and dopaminergic function in addiction-related behaviors. So I'm planning on writing my K99 along those lines.
Yeah, and so I'm very interested in trying to understand the neuroscience of risk and resilience in addiction. So one of my current projects that's funded by a K01 award is a longitudinal neuroimaging study of college students who are at risk for alcohol use disorder. And they're at risk based off of either having a family history of alcohol use disorder, or or having a personal history of adverse childhood experiences. And so that study focuses on impulsivity as a potential mediator between these risk factors and future alcohol use. And I'm also finalizing work on a data-driven analysis of a large neuroimaging data set to identify brain markers of risk and resilience for alcohol use disorder. And so my hope, very similar to what Monica said, is that these findings from these studies will be something that will ultimately be translated to provide treatment targets, specifically targeting, high-risk individuals and people that are at high risk based off of either family history or some sort of environmental risk factor, such as childhood adversity.
Super cool. Well, thank you both so much. It was so great to meet you, and thank you for joining us today. And I just really appreciated learning about this exciting and collaborative project between two excellent co-authors, and how you both, teamed together to write up this incredible study on how dopamine function during it has a role in alcohol and alters the circuitry. So thanks again for joining us, and stay tuned for the next Meet the Author interview, everyone.
Thank you.
Thank you so much for having us.
Thank you.